These are your video notes on the cell membrane. So the cell membrane controls movement, which allows the cell to maintain homeostasis. So if you remember, homeostasis is where the cell has a stable internal environment, or even the organism has a stable internal environment. So the structure of the cell membrane is that it's a phospholipid bilayer. Bi means two. So phospholipid is what is made up right here. And the phospholipid has a head that is hydrophilic. So hydro meaning water, philic meaning loving. So the head of the phospholipid chemical right here is attracted to water, and the tail of phospholipids are hydrophobic, which means they are repelled. So water is repelled by the tails right here, and it's attracted to the phospholipid head. So hydrophobic and hydrophilic are two words that you definitely need to know. So the structure of the membrane doesn't just include these hydrophilic head and tail phospholipid bilayers. There are many other things in the cell membrane, such as proteins, which will act a lot as channels, cholesterol, carbohydrates, other glycoproteins. So there's a lot of things that go into making the cell. Here's a much more detailed picture here. We've got carbohydrates, which deal with hormone receptors. We've got proteins, we've got cholesterol. So there's a lot of things that go into this cell membrane that allow it to be a regulator for what goes in and what goes out of the cell. So the structure of the membrane, we've got proteins that act as channels for transport of materials through the cell membrane in active and passive transport. And this is also involved with reception of molecules and enzyme action. So proteins are important. Reception of molecules is important because that might be like a cellular message, and then enzymes obviously are important because that helps lower the activation energy for reactions that are happening within the cell. Carbohydrates in the cell membrane act as identification sites for chemical messengers such as hormones, which is very important at this stage in your life because a lot of that chemical messaging is happening. And then cholesterols help stabilize the fluidity of the cell membrane. However, the more cholesterol, the less flexible the membrane, which is where you run into if you have too much cholesterol in your system. Then you're going to have problems such as like clogged arteries or strokes or blood clots or whatnot, where you're going to be having some hindrance with too high of cholesterol. So there's types of movement. Passive transport occurs through the natural movement of particles, and this is called Brownian motion. It does not take energy to complete the movement of particles in passive transport. Passive transport things will move with a gradient. So it just simply means when we have a gradient, that means one side has more things than the other, so one side has a higher concentration than the other. The three examples of passive transport that we will study are diffusion, osmosis, and then facilitated diffusion. Active transport is the other type of transport we will study, and this requires ATP, or energy, to pass through the membrane. So it moves against the gradient. So I always like to think about this in terms of thinking about running up a hill. It's probably going to take energy to maybe push a ball up a hill, for example. Whereas if we were to roll a ball down a hill, like with passive transport, meaning moving with the gradient, it wouldn't take energy. Okay, so active transport takes energy. Our examples that we will study are protein pumps, endocytosis, and exocytosis. So diffusion just is the net movement of particles from areas of high concentration to areas of low concentration, and it says to reach an equal concentration or dynamic equilibrium. So it's important that we understand that dynamic equilibrium is meaning that dynamic meaning moving and equilibrium meaning equal, so it's moving equally. So things in nature are never stagnant, so particles are in constant motion. So if you've reached dynamic equilibrium, what's going to happen is just things are going to be moving still, but they will be moving at equal rates. So that's what happens in diffusion. So it happens because, again, the particles are in constant motion, so they bump into the membrane, and if the particles can pass, they will. The more particles on one side of the membrane will equal more bumps, and the more bumps will equal more movement. So this is called Brownian motion again, and that just should make sense. If you've got a high concentrated area, those more particles are going to bump into that cell membrane, and more will pass if they can. Here's a good example of diffusion when we look at like a sugar cube, for example. If we drop a sugar cube in water over time, those molecules will diffuse to that dynamic equilibrium. This would even be a better diagram if we were showing those molecules continually moving right here in equal rates all around this beaker of solution. Here's another great example of diffusion over time. We've got our lipid bilayer. Here's we've got a higher concentration. Here we've got a lower concentration, so things will move into dynamic equilibrium. And this, again, would be even better if we were showing those molecules moving in equal rates about each side of that lipid membrane. Another good example here, you can't have too many diagrams, I don't think, of just molecules moving across a membrane. And it doesn't always have to just be one way. We can have multiple molecules moving here where we're showing like different colors and different sizes. Both will reach equilibrium at some point because they're in constant motion. So the speed is based on the concentration differences or the concentration gradient of molecules in an area. The bigger the difference, the faster the diffusion will happen, and the more net movement we will have. So if we've got our box right here, what we would expect is that these molecules would be moving faster to get to that low concentration. So that's just kind of what this one is saying. So again, the concentration gradient, this is a higher concentration. Think like this is a steep hill, so it's probably going to move faster down that hill to get to that dynamic equilibrium. So all cell membranes are selectively permeable, so not all material can pass through it. Semi-permeable membrane, then, which is what we call our cell membrane, controls passages of substances through the membrane. So what can move are small, non-charged particles, and that's really, really important. So larger molecules and charged molecules have a hard time getting through that membrane, and that's where our other types of transport come in when we start talking about osmosis, facilitated diffusion, and our active transport things. So when we talk about osmosis, osmosis is the movement of water through a selectively permeable membrane, and this movement is aided by a membrane protein called an aquaporin. The aquaporin is a protein specific to water in the cell membrane that allows water to quickly move in and out of the cell, which is important because water will always move to dilute. So I believe that you have this diagram on your notes, and so that's just showing like water going into and out of the cell through this protein channel in the cell membrane here. So water moves differently than any other of the solutes. Okay, it moves to dilute. So important. It moves to dilute the highest concentration of solutes. So water will always move towards the higher solute concentration. So when we say solute, we just mean salts or particles or things there to create equal concentrations. Again, it will move and finally reach dynamic equilibrium. So just again, very important that we understand that water will move to dilute. So when we talk about osmosis, we have three types of solutions when talking about cells. So hypertonic is the first type of solution. And so what it means is that the solution, when it says solution concentration, so the solution outside the cell, if the solution concentration outside the cell is higher than inside the cell, then water will move out of the cell to dilute the higher concentration. Water will then move out, reach dynamic equilibrium, and the cell will shrink. Hypotonic is basically opposite of hypertonic. Hypotonic is that the solution concentration is lower on the outside than on the inside, so that the inside of the cell now has the higher concentration of stuff. Water, again, will move to dilute, so if it's moving to dilute, it's going to move into the cell, and the cell will swell. I like to think that hypo sounds like hippo, and hippos are fat, so hypotonic fat cells, they'll swell because it's gaining water. So just a little wordplay, hopefully that helps. And isotonic, so isotonic solutions, the solute concentration is equal to the cell, so there's no change in cell size, but it is important that we understand that water will still be moving, it will just be at equal rates, so it will be in dynamic equilibrium. So I like this diagram right here, you have this in your notes, because it's showing us pictures of where the water is moving. If the fluid outside the cell has higher concentration, water will move out of the cell to dilute, so then the cell will shrink. So this is just basically what we just had on that other slide, but this is just giving you a visual with it. So if it's hypotonic, the cell will swell. So hypo, hippo, just kind of remember that. And then if it's isotonic, I really like this diagram because it's just showing water moving into and out of the cell in equilibrium there. So to kind of connect to our information that we learned in our organelles, we know that a contractile, we know a vacuole is an organelle found mainly in protists, a contractile vacuole that controls water. So we call it osmoregulation within a cell. So when you guys were looking at your protists underneath the microscope and you were seeing it kind of like sucking in, possibly eating, possibly filter feeding, we were also probably seeing a lot of the contractile vacuoles, and that would be pumping and collecting water from the cytoplasm in the cell and extracting it to the environment. So we probably saw a lot of that contractile vacuole in action, which is pretty cool.